Hi everyone. Oh, Hi everyone. I'm Hazel, and today with me we have Julie. Welcome to the SCSC Club this week. Okay, so for this week, uh, I'm sorry. Oh, we sorry, a little bit technical issues, but it's okay now. So welcome to the SEC Club this week. For this week, as you may see in the screen, we will explore the microenvironment and drug response in refractory multiple myeloma. And for this week, we will explore this cancer using our newer version of B-Browdex, which is B-Browdex Pro. So this is like a newer version for our platform for exploring, investigating single cell RNA-seq data. Okay. Okay. Sorry. Okay. Okay. So before we dive into this week's section, let's give us a little bit of last episode recap by Julie.
Yeah. So from last week, we studied the tumor microenvironment in head and neck squamous cell carcinoma. And when we looked at our population of malignant cells and we clustered by the different patients, we found that the clusters are highly associated with the patient ID, which means that each patient expressed quite a different expression profile in their malignant cells, which means these malignant cells are quite heterogeneous in their phenotype. Whereas in our non-malignant cell population, we found that they clustered by the cell type instead. So given this, we found that the clustering is driven by different factors. In our malignant cells, we found that it's driven by patient ID, whereas in non-malignant cells, we found that they're driven by cell type instead. And then furthermore, deeper into the analysis, we queried some genes that are related to partial epithelial mesenchymal transition. So the three genes down here, SNAI2, VIM, and TGFBI, are all markers for partial EMT. Whereas if you can see, when we start at the root cell or at the primary tumor site, we see that the expression kind of heightens at the early stages of the tumor progression. However, when we look at this one particular gene, KRT14, and we follow that progression, we see that the expression actually decreases as it gets towards the site of metastasis. And this gene is a marker for epithelial cells, which means that these cells may be partially losing their epithelial phenotype. Okay, and then so we were able to link this to metastasis in head and neck squamous cell carcinoma, where partial EMT is initiated at early developmental stages of the cancer progression and may diminish following metastasis.
Yeah. So last week we already explored the microenvironment of head and neck cancer. And coming for this week, we will explore the microenvironment of multiple myeloma. So this is a very popular blood cancer. And today, not only the microenvironment, we also explore a very common case in this cancer, which is the relapse or refractory multiple myeloma. And before we dive into what we have found, first we'll give you a little bit of, of the overview of this disease. Yeah.
Okay, so a little bit about multiple myeloma. It is an incurable and rare cancer that originates from plasma cells, which are our white blood cells that are responsible for producing antibodies. And it's the second most common blood cancer after leukemia. And the mechanism by which this occurs is when the uncontrolled proliferation of malignant plasma cells occurs in this disease, leading to the production of abnormal antibodies called M proteins. And the excessive production of these abnormal antibodies can lead to a whole host of health complications, such as hypercalcemia, renal dysfunction, and anemia. And so one of the most challenging aspects of multiple myeloma that remains is its high relapse rate, with over 16% of patients experiencing early relapse within the first 12 months. Whereas in long-term relapse, 66% of patients relapse within 4 years. So this makes long-term remission quite difficult. Multiple myeloma is also associated with bone lesions and osteoporosis due to increased osteoclast activity, hematopoiesis, and resulting in reduced red and white blood cell production. So although multiple myeloma therapies have significantly improved patient outcomes, this relapse or refractory period remains a significant challenge. And so ongoing research is quite essential to finding more effective therapies. And so that is the basis or the motivation behind why we're researching multiple myeloma today.
Yes, sure. So like based on all of that knowledge about multiple myeloma, today we will explore like a very specific region, which is, as you can see in the figure here, we will actually explore the bone marrow microenvironment, which is the main disease region in this blood cancer. And then for the second part, we will explore like why do we see such a high relapse rate after like only 4 years, like more than half of the patient relapse after 4 years. Okay. So so to explore this disease today, we are looking at one specific example database. So this database or this study is about the subclone-specific microenvironmental impact and also drug response in refractory multiple myeloma, which reviewed by the single-cell transcriptomic, so that to understand the complexity of the bone marrow microenvironment and also the response to treatment of refractory multiple myeloma. Okay, so in this study, the scientists look at the samples from 20 patients. Okay, so they look at sample from 20 patients that have been relapsed from at least one previous treatment at the point of experiment. And after sample were collected, they were sorted in here. They were sorted to identify the CD138 positive myeloma cells before going through the single cell analysis. And okay, and then based on this very specific study, the authors have found the first result, like they have found the gain of chromosome 1q, or like the gain of the long arm of the chromosome 1. So as you can see here, that based on the literature, the gain of chromosome 1q is found in multiple myeloma at all stages, including the newly diagnosed and relapsed or refractory multiple myeloma. You can see on multiple studies, like multiple literature that we found here. So it is associated with worse prognosis, increase the tumor burden, and decrease the overall survival of the patient. And then the frequencies of chromosome 1q gain tends to increase as the degrees progresses, making it a significant marker for assessing disease severity and guiding treatment. So based on all of this literature, we then have the first question for our investigation, which is then because we see the gain of chromosome 1q in so many other studies, so we must see the 1q gaining in this specific study. Yeah.
Right. So here in the paper, the authors were able to striate the gain of chromosome 1q into 3 different levels, into high, intermediate, and low. And then when we compare our high and our low levels using differential gene expression analysis, we see an upregulation in particular genes in our different groups. And then we perform a pathway enrichment analysis, and we see upregulation in pathways such as apoptosis, G2M checkpoint, crash signaling, and myelogenesis, all of which are related to cell cycle, cell fate, and survival. So overall, this suggests that a high 1q positive or 1q gain group is related to the survival of cancer cells or myeloma cells during the response to treatment, and thus related to the relapse of patients.
Yeah. Okay. Then, okay, so in this slide here, so we are showing you some genes that related in this specific purple heat map right here. Like here are some genes that related to the gain of chromosome 1q, and you can see right here that MCL1 is highly expressed in all groups. Okay, so such as a general function in multiple myeloma. But first, let we explain for you like what is MCL1 and what is the function of this gene.
So MCL1 is also known as myeloid cell leukemia 1. It's a pro-survival protein that inhibits apoptosis by kind of sequestering pro-apoptotic proteins, and thus this promotes cell survival. So MCL1 does promote cell survival because it plays an important role in regulating like mitochondrial dynamics, energy metabolism, and cell cycle progression, which contributes to the overall cell homeostasis. So due to its involvement in cancer cell survival and resistance to therapies, MCL1 has become a promising target for cancer treatments with specific inhibitors currently being explored in clinical trials. So one of the ways we can explore this gene, MCL1, even further is by looking at what other genes are co-expressed with MCL1. When we query for it in talk to data, we're able to see that it is lowly co-expressed with some of these genes here. And when we look at the function of these genes, they're related to cell stress, cell proliferation, cell survival, and just overall cell cycle related genes here.
Yeah, so in this correlation heat map right here, these are actually the genes that correlated with MCL1 in multiple myeloma only. So this correlation is specific to the cancer, but not like in in other cancers and or in normal conditions. Yeah. So based on this finding, we then move on to the next question, like because last week we see like a very complex heterogeneity in head and neck cancer, like okay, because we see that in head and neck cancer. So we kind of like suppose that the situation, like the complexity of cell composition will be the same in multiple myeloma. Okay, now we move to so that question leads us to the second part, that is relapse or refractory multiple myeloma is associated with a complex bone marrow microenvironment. Yeah.
Yeah, so in this analysis, we were able to mark for our plasma cells here using these 4 markers, and we were able to kind of select and subset for these cells. So if you remember, plasma cells are the drivers of multiple myeloma when they become malignant. So these are possibly our malignant cells that we mentioned from last week. And then when we color these plasma cells by the different patients, we see that the clusters are highly associated with the patient ID, which means that each patient is expressing a very differentiated expression profile of plasma cells. However, when we look at our non-plasma cells here in the other clusters, we see that they're quite homogeneous by patient, which means that the bone marrow environment is not as heterogeneous or not as complex as our plasma environment.
So yeah, so the situation is basically identical to what we saw last week. Is that in non-malignant cell, in here is like a different cell type in the microenvironment. We can see a quite, it's the same in every patient. So we can see the clustering is mixed together. But however, when you look at the plasma cell or the malignant cells, similar to the head and neck microenvironment, we see that each patient form a very specific clusters, suggesting that the plasma composition is distinguishable across multiple patients. Yeah. Okay. So now let's take a closer look into what cells are really we see in the non-malignant cells.
So in our non-malignant cells or our bone marrow environment, we can see a lot of different cell types, especially related to immune. So here in the composition, we see a very large proportion of myeloma cells. And then other cells that we see large proportions of are CD8 positive or other CD14 positive and NK cells. So this suggests that the immune environment in in the bone marrow is quite complex because we have a lot of immune cells that are communicating.
Yeah, so basically we see a lot of T cells in this bone marrow microenvironment. And then I remember from the beginning that the multiple myeloma has a quite a high rate of relapse or refractory. So I question that these complex pattern, like these complex cell type composition here, must be contributing to that, of the relapse or refractory rate. Is that correct?
Right. So that's one explanation for why we're seeing this complex immune environment.
Yeah. Okay. Okay. So now, based on that, we move on to the next part. It's like, we want to ask whether the complexity of the bone marrow microenvironment really related to the relapse or the refractory rate of multiple myeloma. So that we lead to this part where we will explore further into the treatment response in relapse or refractory multiple myeloma. Okay. Oh, wait, sorry. Okay. Yeah.
Okay, and so the first way we can look at if there are differences in the treatment response is by looking at the cellular composition. Here we're looking at the composition at two different levels. So at this first level, we're looking at like a higher level of the cell type, whereas in the second analysis, we're looking at a more specific or a lower level cell type annotation. So just visually, you can see there's not really much of a difference in the response versus the stable disease between both of our analyses, which means the cell type composition are quite similar between the two. So the cell composition or the cell proportions may not be what is driving the differences in the treatment response in our patients. So furthermore, another way we can kind of look at the differences is by using differential gene expression analysis. So of course, when we compare our response and our stable disease, we see an upregulation and downregulation in a couple of genes. And then to get an overview of these genes, we can perform a pathway analysis. And so, okay, so here on the left-hand side, we're looking at the pathways upregulated in our response group. We see very similar pathways upregulated in both our response and our stable disease. For example, we see interferon response, epithelial and mesenchymal transition, TNF alpha signaling, and also IL-STAT signaling as well. And all these pathways are related to immune evasion, right, which are drivers of cancer. However, when we look at the pathways that are specifically differentiated between the two conditions, we see that the G2M checkpoint pathway is upregulated in our response. And because the G2M checkpoint pathway is like a cell cycle checkpoint pathway, this can kind of be, we can interpret this by saying that uh, the cells are kind of coming to this checkpoint, and then the body recognizes these cells as being malignant possibly, and therefore they are not able to progress further into the cell cycle. And so in that way we see a response, a positive response. However, in the stable disease group, we see an upregulation in apoptosis. And one way we can interpret this is that the rest of these pathways are kind of evading this or escaping this apoptosis pathway because we're looking at our stable disease group, right? Which means that the cancer is still kind of progressing. And so the interplay of all of these other pathways kind of helps escape this apoptosis pathway.
Yeah. Like basically the pathway enrichment situation in response group and the stable disease group are in generally quite similar to each other. Yes. Like, and one thing not really related to this situation here, but what is very interesting here is that we also see the enrichment of epithelial mesenchymal transition again. So if you already watched the previous section, so the EMT is a very common process to occur in the head and neck cancer. And I think like this may show that even in different type of cancer, some common genes always get enriched during the cancer situation, therefore leading to the similar pathway enrichment pattern right here. Yeah. Okay. Okay, and what also very interesting is that even though we see that the two groups have quite the same complexity, like it has the same pathway enrichment set, but finally have very different outcomes. Yeah. So okay. So that like based on everything, because we don't see the contribution of the cell type composition in here, we don't really see the contribution of the pathway enrichment here. So then we come to another way that we hope that can explain the difference in relapse and I'm sorry, in response or non-response group, is that the actual cell-to-cell communication in the microenvironment. So here we actually dive deeper into the communication between the myeloma cell and the bone marrow microenvironment in the hope of like explaining the reason why we see non-response versus response. Yeah.
Okay, and so here I'll dive into the demo on vBrowser X Pro. Okay, so on vBrowser X Pro, we do have one new feature, cell-cell communication, that lets us see what ligand and receptors are at play and what kind of cell types are communicating with each other. So here we're looking at our data set. We're looking at our stable disease versus our response. Let's say I just want to see what cell types are at play or communicating in our response group only. I will select that and I will subcluster that. And then I can click on cell-cell communication. And then you can choose the metadata to run it by. So I want to see the interplay between our different cell types here. And then I can click run. So let me open up a results page so you can see what this looks like. So here we're given a heat map. You see our senders on the y-axis and our receivers on the x-axis. And you can see that hovering over each node, you see the number of interactions and then the sender and receiver cell. So here we see a lot of interactions in the myeloid and dendritic cells, and then a lot of interactions in the T and NK cells as well. And let's go ahead and click on a node, and it'll open up the interaction pair as well as the pathway and some statistics related to that.
Yeah.
So these myeloid dendritic cells, you can see they're upregulated in MHC class 2 pathways, here in MIF pathways, here and here. And then in our T and NK cells, these are more upregulated in MHC class 1, here, MHC and MIF as well.
Yeah.
Okay, so given that, let me go back to our slides to give you a summary. So given that, remember again, we saw an upregulation in MHC class 2 pathways in our myeloid and dendritic cells. And so these MHC class 2 molecules are kind of antigen presenting so that the immune system can respond. But then our MIF pathways, these are more suppressors for these MHC pathways. So in our response group, we see more interactions in myeloid and dendritic cells. However, in our stable disease group, we don't really see that as much. And so given that, the upregulation of communication in myeloid dendritic cells in the response group means that the MHC class 2 pathway plays a role in the response to treatment. But because we see an upregulation in our MIF pathways as well, it's kind of suppressing this MHC class 2 pathway, so which leads to being responsive to treatment.
Yes. Okay. So I hope that like this communication between cell type is quite partly explaining the situation in the relapse group. Yeah. And also, please let me know because we think that maybe the cell-to-cell communication tool is quite new to everyone here. So please let us know if you have any question regarding the how we do that or like the interpretation of the result. Please let us know. you can type in the Q&A and we will get you the answer right away. Okay. And that's also the last result of our session today. So to sorry, to summarize, Julie, can you please, because I believe we have 4 main points to summarize today. Can you please, let us know what we get from the section today?
Yeah, so in the first analysis, we found that the gain of 1q chromosome is associated with pathways such as cell cycle, sulfate, and survival, and this contributes to the resistance and relapse of multiple myeloma patients. And we also see this same trend across other cancers as well. And then in the bone marrow environment, we saw that our plasma cells, which are drivers of our disease, is highly heterogeneous. However, in our non-malignant cells or our non-plasma cells, we see that the expression is not really patient-driven. It may be driven more by cell type or some other type of metadata. And then the third point, we see an increase in communication in myeloid and dendritic cells through the MHC class 2 pathway. And this pathway contributes to the response of multiple myeloma patients to treatment. And then lastly, we also saw that the MIF pathways are activated in multiple myeloma, and this could be one of the mechanisms by which multiple myeloma uses to suppress these MHC pathways and facilitate the immune evasion.
Yes, okay. That's like all of the take-home points for our session today. So now, first, okay. Now, that we are moving on to the Q&A section, where please let us know if you have any question, any point you would like to explain further through the section. And also here, because we are now showing you the newer version of our single-cell analytic data analysis platform, bBrowser X Pro. Please let us know your idea so that we can build a better platform. As you can see, may see the survey is popping up on the screen right now. We are having some questions for you about, which features you will usually use for your analysis. And then like for the cell type annotation tool, what way would you like to use for your annotation process? And this is like the very important thing to do while analyzing single cell analytic data. And then regarding the visualization tool, what is the one that you most most frequently use? And then we have overall just 5 questions here. Please let us know your opinion on this. And yes, please spend some time, answer the survey, and let us know if you have any questions. Okay. And also here, because the holiday season is coming very close. Next week is actually the Christmas. And so that, let me just go here. So that, we will also take a little break for the next 2 weeks. So the next 2 Fridays will be no SEC Club at all. And we will return on the new year on 11th of January. And then before the holiday season, we wish you a, very happy holiday season, like a Merry Christmas. And we also wish you the best, all the best for the new year. Yeah. So please stay tuned a little bit to fill in the survey, and then please just know if you have any Q&A section. And yes, that's all for today. See you next year.
See you guys next year. But before that, for you guys who stick around, I do want to talk a little bit about the roadmap that we have for bBrowser X Pro. So these are the features that we have available in bBrowser X Pro right now. In phase 1, we've released all of these features in blue and green. So it includes that cell-cell interaction feature that we just saw, trajectory inference, and some of the visualization tools as well. However, in phase 2, this is where we get into more advanced analysis. So one of the 2 biggest improvements to bBrowser X is that we support the upload of very large data sets up to 10 million cells. And the second most important feature is our development of more advanced analysis. So this includes gene regulatory network, perturbation analysis, more multimodal analysis, so integrating in ATAC-seq, perturb-seq, and whatnot, CNV analysis, adaptive immune receptor.
Yeah.
So these will come into phase 2 in vBrowser X Pro.
Yeah. Okay. Here, let me see that we have any question. Okay. Oh. Okay. Okay, so oh yes. I think that we got one comment from Wang. The comment is that the MIF pathway role in antigen presentation in not only immune suppression. So in here, Wang, I'm a little bit I'm sorry. I think that I don't really understand the question quite well. So can you please elaborate on anything that you would like to ask related to the MIF pathway? Or Julie, do you have any opinion on this? Like the comment is about the role of MIF pathway in the antigen representation. Ah,
Yeah, I think
yeah.
this is just one interpretation. So he comments that the MIF pathway, its role in antigen presentation is not only does not only work for just immune suppression. It could work in other roles as well. So I think that is worth investigating further to try to find interpretation for the results that we're seeing.
Yes, yes. Okay. Oh, thank you so much. Okay. Okay. So thank you, Wang. We will explore further on another role, multiple roles for the MIF pathway, because when we also understand that one pathway usually not only serve for one role only, but it depends on the result representation. And then we will explore that further. Thank you so much for your comment about the role of the MIF with the MCH2. Okay. Okay. Okay, so we also have another question from Yang about the generation of the data. Also, in here, actually, let me just open the slide again. Oh, I'm sorry. In here, actually, we do not generate the data ourselves, but we rather, this is the actual publicly available data set, and then we can get the data set from this paper, and then we can, bring it back. We have a team to curate all the data. We have a big database for all of these single cell RNA-seq data set. And then once we have all of those data, we can analyze those on our platform, on our analysis platform. So I hope that answer your question. Yes, so this is the publicly available data, not our internal data. Yeah. Okay. Okay, so we done this one. This one. Okay. So do you have any further question for us? Please let's just know. Yeah. Okay, so this is done. Okay, so if we have no more question, I would like to end the session today. And again, we wish you a Merry Christmas and Happy New Year.
Happy New Year, everyone.
Yeah, see you in 2025. Okay, bye.